In this video lecture, we'll see polyps in large intestine and the tumors of the large intestine. Polyps are the most common in the colorectal region but may also occur in esophagus, stomach or small intestine. Polyps begin as small elevation of the mucosa and these are referred to as sessile polyps. That is a term borrowed from the botanists who used the sessile term to describe the flowers and leaves. So when there is a stalk formation in the sessile polyps, then they are not told as a sessile polyps, but they are called as a pedunculated polyp. So two types of polyps, one is sessile and other is pedunculated type. They can be classified also as either non-neoplastic or neoplastic in nature. Most common neoplastic type of polyp is the adenoma, and it has the potential to progress to the cancer. The non-neoplastic types of polyps are hyperplastic polyps, inflammatory polyp, hamartomatous polyp, and lymphoid polyp. Or inflammatory polyps. The neoplastic types are tubular adenoma. So these all are adenomas, tubular villus or tubulovillus types of adenomas. So hyperplastic polyp is the most common polyp of the large intestine. The chronic hyperplastic polyps are benign epithelial proliferations that are typically discovered in the 6th and 7th decades of the life. They are asymptomatic. The most common site is rectum. The pathogenesis of formation is incompletely understood, yet they are thought to result from the decreased epithelial cell turnover and delayed shedding of the surface epithelial cells. Their chief significance is that they must be distinguished from the sessile serrated adenomas that are histologically similar but have malignant potential. Microscopically, they are most commonly found in the left colon and are typically less than the 5 mm. They are smooth nodular protrusion of the mucosa. They are composed of elongated crypts and well-formed glands and do not have any malignant potential. These are hyperplastic polyps. Polyp surface with irregular tufting of the epithelial cells. So these are epithelial cell tufting, and this tufting is because of epithelial overcrowding. Next type of polyp is non-neoplastic. Next polyp is hamartomatous polyp. Hamartomatous polyps occur sporadically or as components of various genetically determined or acquired syndromes. Juvenile polyp is a type of hamartomatous polyp. It is a developmental malformation affecting the glands and lamina propria, commonly occurring in children under 5 years old. In adult, they are called as retention type of polyps. Site is in the rectum, and the most common presenting symptom is painless rectal bleeding. These may be sporadic or syndromic, and the morphology of these two is often similar. grossly large, rounded, pedunculated, smooth surface, histologically mucus-filled, cystically dilated tubules, lined by normal or inflamed mucosa, no malignant potential, or they may occur as a syndrome. Next type of hamartomatous polyp is Peutz-Jeghers polyps associated with Peutz-Jeghers syndrome. They are accompanied by mucosal and cutaneous pigmentation around the lips or mucosa, face and genitalia. They are autosomal dominant in nature, caused by germline mutation in the LKB1. The median age of presentation is 11 years. There are multiple hamartomatous polyps in the gastrointestinal tract and also mucocutaneous pigmentation. They have an increased risk of developing carcinoma of the pancreas, breast, lung, ovary and uterus. So these are hamartomatous polyps. Next type of polyps are inflammatory polyps occurring in the patients with long-standing inflammatory bowel disease, especially in the chronic ulcerative colitis, and they are usually multiple. So they may be a part of a solitary rectal ulcer syndrome also, and these examples of that is purely inflammatory lesions. And the solitary rectal ulcer syndrome patients present with the clinical triad of rectal bleeding, mucus discharge, and inflammatory lesion of the anterior rectal wall. And the underlying cause is impaired relaxation of the anorectal sphincter that creates a sharp angle in the anterior rectal self, and that leads to recurrent abrasion and ulceration. And there is a chronic cycle of injury and healing in the inflammatory polyps with solitary rectal ulcer syndrome. The distinctive feature of inflammatory polyp is mixed inflammatory infiltrate, erosion, epithelial hyperplasia, and lamina propria, fibromuscular hyperplasia. So these all is seen in this picture with inflammatory infiltrates, mild epithelial hyperplasia, and the vascularization is seen here. Lymphoid type of polyp is another type of polyp that is due to reactive hyperplasia of the lymphoid tissue in the intestine, microscopically composed of lymphoid follicles, germinal centers in the mucosa and submucosa of the intestine. So these are lymphoid polyps. Now neoplastic polyps or adenomas. One is adenomatous polyp. This neoplastic mass lesion in the GI tract may produce mucosal protrusion or polyp. The most common neoplastic polyps are colonic adenomas, which are precursors to the majority of colorectal adenocarcinomas. Adenomas are intraepithelial neoplasms that range from small, often pedunculated polyps to large sessile lesions. And there is a small male predominance, and they are presenting with approximately 30% in adults living with by age 60. These adenomas are divided into three types: tubular, villus and tubulovillus. Tubular means made up of tubular glands. Villus made up of finger-like or villus projection. Tubulovillus mixture of tubular and villus pattern. And these colorectal adenomas have presence of epithelial dysplasia. So these are characterized by epithelial dysplasia. Epithelial dysplasia is present in all adenomas. Morphologically, grossly, polypoidal growth usually less than 2 cm is sessile, and if more than that, then they are pedunculated. They consist of tubules glands lined by columnar cells surrounded by fibrovascular stroma. Polyp is covered by columnar epithelium. Tubular cells may show severe dysplasia and invasive carcinoma also. So these are tubular adenomas. Tubules are formed over here, multiple projections of tubular nature. Second type, villus adenoma, least common and largest and most ominous of epithelial polyps. Typical age of presentation is 60 to 65 years. 75% of them is located in the rectosigmoid area. They present with rectal bleeding, anemia. Large ones may secrete copious amount of mucoid material rich in the protein. So grossly, velvety cauliflower-like growth, and they are generally sessile. Microscopically, finger-like villus processes are there. Villi have thin core of fibrovascular tissue and degree of dysplasia with frank invasive carcinoma in up to 40%. These are finger-like projection. Tubulovillus adenoma, they are intermediate in between these tubular and villus adenomas, frequently having stalk dysplasia and malignant potential. So this probability of carcinoma occurring in the neoplastic polyp is related to the following three different things. One is size of the polyp, second is the relative proportion of its villus features, and the presence of cytologic atypia or dysplasia. How much dysplasia is present in the neoplastic cells? Minor amount of dysplasia is there in every polyp. Familial polyposis syndrome or familial adenomatous polyposis, it is an autosomal dominant disorder, familial adenomatous polyposis, and in which patients develop numerous colorectal adenomas in teenage. It is caused by mutation in the adenomatous polyposis coli gene, APC gene. At least 100 polyps are necessary for a diagnosis of classic familial adenomatous polyposis, but sometimes as many as several thousands may be present. And the colorectal adenocarcinoma is there in almost 100% of untreated familial adenomatous polyposis patients, and these occurring before age 30 and nearly always by age 50. Second is Gardner syndrome, in which polyposis coli is there, multiple osteomas are there, epidermal cysts are there, and fibromatosis. Turcot syndrome consists of polyposis coli, gliomas, and fibromatosis. So this is adenoma to carcinoma pathway, normal adenoma and carcinoma. And these are the genetic Different genetic pathways of carcinoma progression: APC loss, KRAS mutation, chromosome 18 loss, and P53 loss. Familial polyposis syndrome. Second type of hereditary type of colorectal cancer is hereditary non-polyposis colorectal cancer, HNPCC, also known as Lynch syndrome. It was originally described by based on the familial clustering of cancers, and there are different sites involved like colorectum, endometrium, stomach, ovary, ureters, brain, small bowel, hepatobiliary tract, pancreas, and skin. It is thought to account 2 to 4% of all colorectal cancers. So it is the most common syndromic form of colon cancer. And HNPCC is caused by inherited mutation in genes that encode proteins responsible for the detection, excision, repair of errors that is occurring during DNA replication. These are different risk factors for development of adenocarcinoma: age, adenomas, polyps, sedentary lifestyle, diet, obesity, family history, inflammatory bowel disease, hereditary syndromes, refined carbohydrates, alcohol, less number of dietary fibers, vegetables, fruits, antioxidants, calcium, and folate is associated with colorectal carcinogenesis. Adenocarcinoma of the colon is the most common malignancy of the GI tract. It is a major cause of morbidity and mortality worldwide. In contrast, the small intestine, which accounts for 75% of the length of the GI tract, is very uncommon site of benign and malignant tumors. So large intestine is the most common site of malignancy as well as the benign tumors. Adenocarcinoma and well differentiated neuroendocrine tumors are two most common types of tumors in the large intestine, and the less frequent ones are lymphomas and sarcomas. The dietary factors most closely associated with increased rates of colorectal cancer are low intake of refined carbohydrates, low intake of these unabsorbable vegetable fibers, dietary fibers, and high intake of refined carbohydrates, intake of alcohol, saturated fat, consumption of red meat. There are different pharmacology. Chemoprevention is also implicated in the modification of colorectal carcinogenesis progression. Dietary modification is also there, but there is one pharmacological chemoprevention, like with the help of aspirin or other NSAIDs, that inhibits the enzyme cyclooxygenase 2. The cyclooxygenase 2, COX2, is expressed in 90% of colorectal carcinoma, and inhibition of that is very helpful in the pharmacotherapeutic prevention of carcinogenesis progression. So this is adenoma carcinoma sequence: normal colon, mucosa at risk, adenomas, and carcinomas. And these are two hit therapy, two hit pathogenesis. So first hit and second hit. First hit is germline or somatic mutation of cancer suppressing gene, and the second hit is inactivation of normal alleles. or methylation abnormalities. So there are two different pathogenetically distinct pathways are there for the development of colon cancer. One is APC beta-catenin pathway in 85% of the cases. Second is DNA mismatch repair gene pathway. Morphologically, the adenocarcinomas are distributed approximately equally over the entire length of the colon. Tumors in the proximal colon often grow as polypoid exophytic masses. Carcinomas in the distal colon tend to be annular lesions. Left-sided carcinoma are annular lesions. Right-sided carcinoma are polypoid and fungating masses, and the obstruction is uncommon. Mucinous adenocarcinoma secrete abundant mucin that may dissect through the cleavage planes in the wall. So here are the pictures of left-sided and right-sided colonic adenocarcinomas. So these are signs and symptoms. If located closer to the anus, change in the bowel habit, feeling of incomplete defecation, and pararectal bleeding. Right-sided lesions are more likely to bleed, while left-sided tumors are usually detected later and could present with bowel obstruction. Serum levels of carcinoembryonic antigen are related to the tumor size and extent of the spread. So these are helpful in monitoring the recurrence of tumor after resection. The Duke classification is used for staging of colorectal carcinomas. Spread of the colorectal carcinoma is the direct spread, lymphatic spread, or hematogenous spread. AJCC or American Joint Committee on Cancer Staging is also a very useful staging mechanism or classification for adenocarcinoma of the large intestine. So microscopically well differentiated, undifferentiated, or moderately differentiated carcinoma is a classification. So these are important prognostic factors for chronic adenocarcinomas. One is histological type, second depth of invasion, third is presence and absence of lymph node metastasis. So this is depth of invasion, stage 0, 1, 2, 3 and 4. So accordingly the depth, if it penetrates submucosa, mucosa or muscularis propria, the stage is here. We give according to the depth of invasion. So in summary, in this lecture we have seen benign and malignant proliferative lesions of the colon, the polyps, neoplastic one and non-neoplastic ones, and colorectal adenocarcinoma. Thank you.